Hi everyone, I'm Viola Hojai and I'm a PhD candidate in UMC Utrecht working with data quality assessment. I will be talking today about the real-world data quality pipeline that containing more than 630 quality checks created as part of the Conception IMI project. The use of real-world data is becoming increasingly important as it allows for more accurate and reliable analysis, but also presents massive challenges, such as different data collection processes, storage procedures, but most importantly, it is data that has not been collected with a study question in mind. And in order to be able to generate valid and trustworthy evidence, first we need to assess the quality of this data. Next, yeah. So what do we mean with data quality? Here's a quote from Juran et al. High quality data are data that are fit for use in their intended operational decision-making, planning, and strategic roles. But does real-world data comply to all of these quality dimensions? We don't know. So this is why we need to quantify the quality of the data before conclusions are drawn. And this leads to the next question. Why do we perform the quality checks? So first of all, we are working with a common data format, which is called the conception CDM. The first step is that we transform the raw data from the conception. So we do the transformation from the raw data to the conception CDM through an ETL procedure that is performed locally. ETL means extract, transform, and load. So the DAPs first identify and extract the relevant data. Then they transform it into the conception CDM by applying rules and finally load it into the target database. So the ETL is a very crucial process and should be checked carefully. So one of the first objectives of the quality checks pipeline is to assess whether the ETL process was performed successfully. Next, as the name of the pipeline, we assess the quality of the data by determining different quality indicators, which we will discuss further on. And finally, we assess whether the data is fit for purpose based on the study requirements. We call it a pipeline as it involves a series of steps that help to identify and address data quality issues from data ingestions to data validation and analysis. Next, the real-world data quality pipeline involves three main steps and one sub-step, which is level 1B. Level 1 assesses the compliance of the data with the conception CDM, meaning the ETL successfulness in this case. Also, frequency analysis and distributions are constructed. Furthermore, we can assess the completeness of the data in this output. Level 1B is a high-level characterization of the data source, which allows for checking the data reliability and the study-specific determinants or the study variables. In level 2, we assess the temporal plausibility of the data by looking at records outside observation periods or records that are happening before the birth date of the subject, etc. And finally, in level 3, we can assess the fit for purpose by looking at counts and incidence rates of diseases or medicine use, vaccine exposure, and pregnancy. Also, we can check the distribution of follow-up times. The quality checks assessment is an iterative process, and all of these steps need to be repeated until the required quality is achieved by correcting identified issues, or when this is not possible, the limitations should be noted. So this brings us to the last step, which is the sign-off. And the sign-off is a document where all the identified issues are explained and has the signature of both the data access provider representative and the quality revisor. Only when the sign-off document has been cleared, then the data access provider can continue with the next steps of the study. If you want to know more about the data quality pipeline, you can use the link shown here to go to our organizational GitHub page. So to make it easier and more clear to how to interpret the results of the quality checks, we are presenting you with a case study. And here we have our case study is that we are interested in evaluating the utilization of ibuprofen during the third trimester of gestation. So our inclusion criteria are pregnant women aged 15 to 55 years old at start of pregnancy, observed during a time period of 2010 till 2020, and without a previous diagnosis of chronic pain 1 year prior to pregnancy. Through the level 1 check output, we can assess the conformance to the conception CDM, meaning the ETL successfulness, the element presence of variables, vocabularies. Also, we check the completeness of the data by using the missingness analysis and also the plausibility of values. In level 1, we have 7 steps with a total of 350 6 checks. Let's start with our case study. To create our study population, we need 2 tables, which are the persons table and the observation periods table. In the first table, we have data about the person, identifier key, about the birth and death date, and sex of the subject. In the second table, we can find information about the start and end date of observation for each of the subjects. In our study, we are also looking at ibuprofen utilization, which means that we need prescriptions or dispensing of ibuprofen. And this information is stored in the medicines table. We also want to know whether the women had a diagnosis of chronic pain. And to retrieve this information, we need the events table, where the diagnostic information is saved. So to check the presence of our mandatory tables, we can use the metadata HTML report produced by Level 1. Next, we will check the conformance of the data by using the HTML report step 1 to 3. In this report, we are able to check whether our tables of interest are formatted correctly, whether the vocabularies are consistent, and also we can check the formatting of date variables. After this assessment, we will see in the output a report called step 4 to 5, which is produced for each CDM table separately. In our case, the example here is given for the persons table. Because we are interested in pregnant women, we have to check that our data source contains sex equal to female. The last step is to check whether the missingness of the variables we need from each of these tables. And we have an example here, which is the medicinal ATC code that we need to identify ibuprofen. Next, please. Next, we have level 1B, and this is a high-level characterization of the data that helps the programmers and also the principal investigator of the study to evaluate the semantic coherence and the precision of the data. It is performed for each table separately by giving all possible combinations of variable values for each table. In the output of level 1B, we can access examples like negative values for numeric variables, such as number of dispensed medicinal products. We can check the presence of diagnostic vocabularies of interest to create our code list for chronic pain. And also we can assess the ATC code to the 5th level to see whether we can actually identify ibuprofen in the data source. Next, in level 2, helps us to assess the temporal plausibility of the records and also the uniqueness and verification of the data. It contains 8 steps, and in total there are 26 checks. 2 very important steps in this level are report 3 and 4. In report 4, we see whether all the persons in the CDM tables are present in our persons table. And because we will construct our study population by using the persons table, all records for the subject that appear in the other tables will be excluded, which leads to a loss of valuable information. The same reasoning can be applied to the report number 3, which is the observations outside observation periods. So if we are looking for a person for a period from A to B, and there is an event found before A, we are excluding that record. So it is very important that the observation period is constructed correctly. Next, we go to level 3. And by using level 3, we are able to assess the reliability of the data, the coverage, and the logical constraints. It includes 9 steps with more than 249 checks, depending on your study requirements. The level 3 reports are quite extensive because their goal is to facilitate the fitness for purpose evaluation of the data sources. For example, we can check whether ibuprofen ATC codes are available in the data source. We can also check whether there is sufficient dispensing of ibuprofen during our study period, which is 2010-2020, to fit our study population, which means that we are able to perform power calculations. And also, we can check whether there are outliers in our pregnancy occurrence or ibuprofen dispensing using the incidence rates. Overall, you are able to check counts, distributions, incidence rates, and prevalence rates in the study population and also in the subpopulations, for example, the drug utilization in pregnant women or subject with a disease. The real-world data quality pipeline has been already implemented in other research networks, such as VAC4U and the EU Pharmacoepidemiology and Pharmacovigilance. VAC4U is a pan-European research network for collaborative post-authorization vaccine safety and benefit monitoring. If you want to know more about them, you can use the website shown here. And the EU Pharmacoepidemiology and Pharmacovigilance is a public academic partnership coordinated by the University of Utrecht in the Netherlands and has been receiving support from EMA. Data quality is key to data-driven regulation. So EMA has recently launched the data quality framework to ensure the integrity and the reliability of data. This framework includes 5 main data quality dimensions, which are coherence, extensiveness, reliability, relevance, and timeliness of the data. Each of these dimensions can be mapped to the conception data quality pipeline. Coherence of the data can be assessed in level 1 and level 2, extensiveness in level 1, reliability in level 1, 2, and 3, and relevance and timeliness can be assessed in level 3. Next. Stop. Yeah. Since the quality checks pipeline is an example of a distributed analysis, meaning that the scripts are sent to the data access provider and then run locally, but they are also quite new and in need of improvements, we are faced with a few challenges. First, the level checks are extensive as well as detailed, which leads to a lot of time to review. Next, adaptations to the scripts based on the project needs and databases particularities might be required. Also, certain hardware is required to run the several scripts that compose this pipeline. We work with R and RStudio, so these programs need to be downloaded and installed. The central processing unit, the RAM memory, the computer storage will highly influence the running time of these scripts. And also, each step is connected to their subsequent step. There are still a lot of challenges to overcome when we work with real-world data, but ultimately, high-quality data is vital for informed decision-making, reliable insight, and accurate predictions. By prioritizing data quality, organization can gain a competitive advantage and drive better outcomes. Thank you.